Okay, so I was asked to sort of review the biochemical and the genetic testing that is done to give you a sense. And what I talked about in the first slide is that the deficiency is here at this level of the 7-dehydrocholesterol reductase. So when you do biochemical testing, the definition of SLOS biochemically is an elevated 7-dehydrocholesterol level. And as I mentioned earlier, cholesterol can often be low, but it can be normal. Okay, the normal laboratory cholesterol test is not sufficient for diagnosis. Okay, it does not distinguish between these different sterols, the 7-dehydrocholesterol and cholesterol. It's some combination of total sterols is what you get out of that test. To test for Smith-Lemli-Opitz syndrome, you need to use, predominantly what's used is a test that relies on what we call mass spectrometry. If you look at these two molecules, the only difference is this double bond, and that means that 7-dehydrocholesterol has 2 less hydrogen atoms than cholesterol, and hydrogen atoms weigh 1 unit, 1 Dalton. So you need to detect the difference between 386 Daltons, that's the weight of cholesterol, and 384 Daltons. And mass spectrometry is the way that that is normally done. The mass spectrometrist instrument weighs the different molecules, and it actually breaks them apart so you can identify what sterol you're looking at. But it relies on this weight difference of 2. So it's specialized testing. It's not your regular laboratory cholesterol test. I just pulled out some examples. This is a report from the Kennedy Krieger Institute. And what elements you can see here, that you have your patient and sample information. If you ever get a report, make sure it is your child and not some other child. I haven't had that happen from Kennedy Krieger. They're good, but I've had that happen on other tests. The report from Kennedy Krieger describes the method, and they're just describing the method. They use what's called gas chromatography mass spectrometry. They measure the weight differences. And then they identify the 7-dehydrocholesterol, and this is the result here. 7-dehydrocholesterol is 90 micrograms per mil. The laboratory also provides what they consider to be the normal range, and for the KKI laboratory, the normal range is 0.16 plus or minus 0.09. The plus or minus just means 1 standard deviation. I'll take you back to school. 68% of individuals fall within 1 standard deviation, 95% within 2 standard deviations. So you see that 0.35 is really becoming their upper limit of normal here. So this is 500 times normal. It's clear that this individual has Smith-Lemli-Opitz syndrome, and that's what the interpretation down here is. This just happens to be a known individual, so they report it out as follow-up. The KKI lab also reports out cholesterol, and you see it's 133 milligrams per deciliter. They also tell us, and I'm not going to talk about it, 8-dehydrocholesterol, but I don't think it comes out on their normal laboratory reports other than ones they send to us. Whoops. Important thing to note, the units are not the same between these two metabolites. The 7-dehydrocholesterol is in 90 micrograms per mil, and the cholesterol is in milligrams per deciliter. Deciliter is 100 mils. It's a weird unit, guys, only used in the United States. But it's so entrenched in medicine that it continues to be used. So if you want to compare the two numbers, you need to convert them and do the mental gymnastics, and believe me, how many times we get this backwards or forwards. But 90 micrograms per mil converts to 9 milligrams per deciliter. So the 9 and the 133 are in the same unit. So it's not like you have 90 units and 133 units in the same units, okay? So the 7-dehydrocholesterol level is much lower, more than a tenth lower than the cholesterol. So that's the Kennedy-Krieger report. There are multiple labs that run this. The other one that we have frequently got reports from is Mayo Laboratories. Mayo Laboratories is this big conglomerate that just seems to have it's easy to send tests to Mayo. And this is an example of their report. Basically the same, but looks a little bit different. They report out a normal range of, they just state less than 2 micrograms per mil. Different ways to indicate this. And their result is 567 micrograms per mil, and they interpret it as either positive or negative. So a report just comes out that this is a positive report. This individual has Smith-Lemli-Opitz syndrome. And then you have Canada and the rest of the world that use what are called International System of Units, or the SI units. Talk about mental gymnastics on the different units. This is mental gymnastics for me. The cholesterol gets reported out in millimoles per liter, and 7-dehydrocholesterol in micromoles per liter. And so the numbers look different. The 7-dehydrocholesterol is reported out as 70 micromoles per liter, which converts to about 25 micrograms per mil in the numbers that I'm more familiar with. And you've got this conversion thing that I always have to look up if I have to do this, so I don't expect anybody. Interesting here, instead of reporting out a normal range, they just say, if you got a positive, call Dr. Novacek to see what to do. Okay. Okay, that's the biochemical testing. Genetic testing is becoming more prominent. I think I'm going to talk about 2 types of genetic testing. One, sequencing the SLOS gene itself. So I say, I think this child has SLOS. I send it off and I say, could you send me back the sequence of DHCR7? What are the variants or the mutations? And so these reports come out, they'll be all different. There are plenty of different groups that run this sequencing. We used to do it when we first cloned the gene. We don't do it any longer. But this is just one example of a report. And what they will do is tell you what their result was. It says the individual is heterozygous. That means the 2 copies of DHCR7 are different, okay? And in this case, they had the IVS8 -1 G to C, that common splicing allele, and then they had a arginine 352 tryptophan, one of the missense alleles, but not necessarily a common one. They can tell it's heterozygous because when you did sequencing this way, it's called Sanger sequencing. What you can see here is you have individual peaks corresponding to the letters, and you read it, and then you get to this peak that's double, and it tells you that there's 2 copies there, one with a misspelling, one with the proper spelling. The alternative is that they could come out and say it's homozygous, and that means instead of 2 different alleles, the child has 2 alleles that have the same change in it. And in that case, you don't get this double peak, you get a single peak, but you can see the misspelling. More commonly now, and especially if somebody doesn't know what your child has and is not thinking about Smith-Lemli-Opitz syndrome, they will send out what's either called exome sequencing or becoming more frequently what's called genome sequencing. What's the difference? Exome sequencing, you're sequencing the exomes, those coding parts of all the genes, and genome sequencing, you're just sequencing everything. And it's just gotten, the technology has advanced to where it's basically more efficient for us just to sequence everything. The reports look a little different. This is just a report from the group we use at the NIH. The primary finding, it says the variants related to the clinical presentation detected. We already knew this child had SLOS. We're just doing genomic sequencing as a research tool. And I'll come to this later, secondary findings not detected. So the primary findings, what they're reporting out here, is that you have this trip 151 termination. That was that common nonsense allele I showed you earlier. And then you have another missense allele. They will make an estimate of the pathogenicity. This is known to be pathogenic. This is very likely to be pathogenic. And then secondary findings, there's an ACGME list of 81 genes that if you do genomic testing, I think it's still 81.
It's about to go up.
Oh, okay. It keeps changing. It was. These are genes that are pathogenic or likely pathogenic and actionable, meaning if I find a variant in it, a reasonable person would probably want to know about that and could do something about that. One could use breast cancer. If I found a breast cancer gene, you might want to know about that in your family. Okay, and I think that's it.
Great, thank you very much.